Hi everyone, my name is Stella Petzova, and I worked with my mentor Matthew from UC Berkeley to use bioinformatics tools to study effector-triggered immunity genes in wild and domesticated tomatoes. I first became interested in this project when I was really young and I was exploring different plants in my area, and I came across two raspberry plants. One of them was clearly wild, it had a lot of thorns and stuff, and had really small fruits, and the other one was domesticated and it had no thorns, its leaves were very smooth, and it had really big and juicy fruits. And from then on, I became really interested in all things evolution and especially plant evolution. And just the development of agriculture also really interested me. And this curiosity led me to study some families of immune receptor genes, which are part of this effector triggered immunity response. That's crucial for helping plants detect pathogens and trigger defense responses. So for my research, I compared two species, Solanum lycopersicum, the domesticated tomato that we eat every day, and Solanum pimpinellifolium. So lycopersicum was selectively bred for size and taste, but at the same time, it lost a lot of genetic diversity, through the process of domestication. On the other hand, pimpinellifolium is this tiny wild tomato that's found in Ecuador and Peru, and since it's never been domesticated, it has a broad range of natural defenses against disease. To analyze this, I compared the gene families, two sets of families in both species. My findings showed that pimpinellifolium has more immune genes overall, making it more robust against pathogens. It also has fewer paralogs, meaning there's less redundancy in its genome, and it doesn't show the same long deletions that I observed in the domesticated tomato. All of this suggests that the wild tomato has preserved stronger, more efficient immunity compared to the cultivated one. This matters because domesticated tomatoes today are under threat from emerging diseases that specifically target effector-triggered immunity response. Without sufficient immune diversity, crops are much more vulnerable to outbreaks, and many crops in the past have been wiped out, such as a species of banana that we no longer have because it was all wiped out by disease. And that's due to their homogeneity and lack of immune response. The applications of my research go beyond just understanding the differences between two types of tomato. By identifying the specific genes that have been lost in domesticated tomatoes, breeders and genetic engineers can work to reintroduce or enhance them in domestic populations. This could help create tomato varieties that are not only productive, but also resilient to future disease pressures, helping protect a staple food for millions of people worldwide. Thank you.